My title is a little bit different. Actually, it's a lot different, but the subject matter is quite similar. The title of my talk is Physics, AI, and the Environment. As Melissa mentioned, I'm from Harvard Medical School in the Computational Health Informatics program at Boston Children's Hospital. So we've heard a lot of great talks, I think, about the application of machine learning to toxicology, environmental health questions. And at the risk of oversimplification, I think there's a couple of common patterns that we see in very successful applications of machine learning. And these are certainly not unique to environmental health or exposome questions, but they are lots of clean labeled data. Good example outside of today's group is ImageNet out of Stanford. Convolutional neural network development has been accelerated greatly by the existence of that database for the community. Efficient compute in time, the best example, and this is skewed towards deep learning, is the use of GPUs, graphics processing units, in recent years. And then I think we've had algorithmic advancements for several decades that have really taken off over the past few years, but we've also had some really amazing breakthroughs over the past couple of years. Dropout is a good example for deep learning. And so the question I like to think about is, do we have these ingredients for questions that we all care about today in environmental health and for studying the exposome? So this is work I did with a very close colleague, Chirag Patel, and it just underscores to me, I think, a fact that many of us know in this room, which is that these exposures that we care about are very complex. They're densely correlated, they're time varying, they're often very difficult to measure. We looked at 575 exposures from the Haynes database, looked at the pairwise correlations, and you can see in red are positive correlations. There's many correlations between these individual factors, one with another. Many of these are statistically significant. They survive replication in separate cohorts of the CDC Haynes data set. And this creates, I think, this big soup of correlations that we really have to tease apart for causal inference. This is a nice, it's a schematic or depiction of just another aspect of this complexity. I mentioned that they're time varying, so they can have very different timescales of exposure throughout life. You can have cumulative exposures, cadmium, PCBs, that accumulate over the entire lifespan. Really large exposures in utero that are very short-lived relative to the lifespan, but potentially very, very influential. Constant but excreted phenols, vitamins that maybe start around adulthood, interventions in the form of drugs, allergies that are seasonal, things like this. So, I'm going to jump into comparisons to genomics, but it was mentioned earlier when we're thinking about looking at A's, T's, G's, and C's compared to trying to study this soup of signals, we're obviously up against a pretty big challenge. So I think we've seen actually some very convincing examples of AI and machine learning techniques applied to questions in environmental health. And I think if we look outside of environmental health, we've also seen some extremely impressive results in medicine more broadly. Image recognition comes to mind, speech recognition. These are tasks where we have a lot of data and we have some very, very impressive published examples. But I'm going to argue that, while in many of those domains, out of the box, these techniques work pretty well, it's unlikely that in environmental health and exposome that these same techniques are going to work out of the box. I think there are pervasive issues around measurement and reproducibility that are likely to limit application. So reproducibility is going to be a central theme of my comments today. Of course, in science, generally reproducibility is in many ways the central thing that we all care about. The great 20th century philosopher of science, Karl Popper, said, non-reproducible single occurrences are of no significance to science. And maybe a more contemporary philosopher of science, John Oliver, devoted an entire segment of his pretty popular show on HBO to scientific reproducibility, and defined p-hacking actually on the show as basically collecting lots of variables and then playing with your data until you find something that counts as statistically significant but is probably meaningless. And so I think that's a pretty reasonable definition of p-hacking. So I thought, to make this a little more concrete, we could take a deeper look by starting with a very powerful machine learning method for the exposome, and that's linear regression. And so I will defend regression as still an incredibly useful technique for most of what we do. So it gives us a couple of things that many techniques do not give us. One is variance explained in the dependent variable that we care about. We can debate this, but readily interpretable coefficients. It gives us predictions. It gives us uncertainty both about our inferences as well as our predictions. And so, it's just one example. Suppose we want to associate lead exposure and C-reactive protein, which is of course a blood marker for inflammation. That's also very clinically significant. So. Haynes is mentioned a few times today. National Health and Nutrition Examination Survey is publicly available. A wonderful data set that comes online every 2 years as a new cohort. So we use this data set probably the most of any single public resource in our analyses. This is a couple of lines of our code for how we can actually execute this analysis. We could read in that data frame that's stored on my hard drive as an Haynes object. I could build an unadjusted model. It's a single line of our code to build a linear model that relates CRP to lead exposure. Supply that data frame there. And I ran this yesterday with a randomly chosen subset of the data, and I got a p-value of 0.12 from that analysis. So then, the first thing you might notice is that model is not adjusted for anything. It's an unadjusted model. Okay, I know sex and BMI are very important variables to mediate many of these relationships. So I could try just sex as a factor variable on the right-hand side there, BMI as well. Well, then race and socioeconomic status are also super important, but then maybe smoking, but not with socioeconomic status. And the list goes on and on and on with how many models I could try here. And eventually, when I did just a couple of these in RStudio yesterday, I got to a p-value of 0.053, which maybe I would describe, or actually I wouldn't describe this, but it could be described as trending towards significance, which I think might make some of you cringe as a term. Then I could have my aha moment and I could filter, right? So one thing I haven't permuted yet is my filtering criteria for who's actually in this regression. So a little bit of tidyverse, one line there. I could filter my age criteria to just 26 or above 25, less than 35. And suddenly, p-value less than 0.05. I could publish, and academic rewards will follow. You can imagine step 5, step 6, step 7, many ways that we can actually create these models. And this is just linear regression. I haven't done anything super fancy yet. There's many analytic choices that I have in even building a model like this. So, very close colleague, Chirag Patel, studied this, formalized this, and studied it at scale with what he called the vibration of effects. And so the idea here is to take this set of covariates that we typically adjust in these models. So they looked at 13 variables in the Haynes data set that are often used as adjustment variables for looking at the relationship between exposures and quantitative phenotypes. Those 13 variables, then you have a choice of whether to include or not include each of those variables. So it creates a set of 2 to the 13th models that you can specify for each exposure phenotype relationship. So that full set, then power set, is used to create each of those little dots on one of those plots that you see that describes a relationship, the quantitative relationship between an exposure and a phenotype. So one example is vitamin E and mortality. We have a Cox regression model, and we look at the actual hazard ratio attached to that exposure and mortality. And so you can see from the volcano plots here, so the minus log 10 p-value is shown on the y-axis, and the effect size is shown on the x-axis. First, relative to to any individual model, there's a whole space of models that you can always build that produce this very large range of effect sizes for each of these relationships. And for many of these actual exposure phenotype relationships, the sign of the effect flips. And so they term this the Janus effect for the two-faced Greek mythological, looking forward, looking backwards reference here. So I first saw this paper a few years ago. Actually, I think independently, and then we've been adopting this approach, but looking at other questions. And I'd say, in some sense, it was both an amazing paper and it also scared me quite a bit because it really puts into, I'd say, an empirical assessment how much flexibility we have when designing these models. So when we're applying machine learning, we have even more analytic choices. We have all those same choices, right? We have inclusion criteria. We have which covariates we care about. We have what data we use. But then we also have a lot of very interesting machine learning specific, in some ways, covariates. And again, this is a little bit biased towards deep learning, but one example is hyperparameters. So I can set my learning rate. It's actually incredibly important to set that correctly to train properly, but there's still a lot of freedom in doing that. Model architecture, so the number of hidden layers I use, the number of hidden neurons per layer, those are all choices the researcher can make. The declaration of improvement, so I actually think this is, it's just the third point there, and it might get kind of blended into the rest of these, but this might be the most important point on the slide. So, no one is going to debate when there's a 20% reduction in ImageNet in a benchmark by Jeff Hinton that is not a reliable and a significant improvement in classification of images. But when we are typically reporting results in our community and in many medical applications, we are often seeing things like 0.01 improvements in AUC in a binary classification problem, maybe even in the hundredth decimal place. And it's been mentioned several times already. I think it's a very non-trivial task to say that is a reliable improvement in AUC. There's at least several decades old literature around quantifying statistical significance of AUCs. But I'd love to talk about that more because I think it's very, very up for debate how to do that correctly most of the time in practice. Splits, so I could decide that training, val, test splits are the right way to approach this, or do cross-validation. Both are readily justifiable. And then another point, which I think is subtle, but also very relevant for the problems that we consider, are Kaggle many analysts analyzing the same data set. And so one example of that is a Kaggle competition. So there's typically a public leaderboard, there's a private leaderboard, and to some extent this process gets corrected out that way. But when we deal with problems where we have large shared databases and many of us are choosing to publish, there's this interesting thing that we've noticed and that we've started to formalize now, is that in some high-throughput investigations, we almost always formally address multiplicity. But when we have a large shared database where thousands of investigators are simultaneously mining the shared database, we very rarely formally address multiplicity across those investigations. So the question is what can we do to potentially improve the situation or make sure we're doing reproducible, robust science going forward? So there's at least two fields, and I think there's some very good examples in both genomics and physics, hence the title of the talk today that I'll talk about. So the first is human genetics research. So there's a major study design change that took place maybe around the mid-2000s. It really got started, which is the transition from candidate gene era studies to genome-wide association studies. And I think Professor Mukherjee has mentioned GWASs. It's been mentioned a couple of other times. So this is really a pretty big transformation of how genetics is done. GWASs have become standard now. Previously, and still to some extent, there's many of these studies. There was, we would investigate one locus or one gene at a time for its relationship with a particular phenotype or disease. And then there's a pretty big switch following the Human Genome Project, HapMap, some other projects that let us take an agnostic, comprehensive view of association of different loci across the human genome with a particular disease or trait. And on top of this, there's been a lot of work in correcting for confounding. And one of the main sources of confounding there is ancestry differences between the cases and controls. And so I should have included a picture or two to show that, but this review does a pretty good job of describing that change by Peter Vischer and AJHG. So we've been inspired in our work to take GWAS and human genomics-inspired approaches in looking at exposures. And so Chirag Patel had come up with the EWAS, which is the sort of direct analog, the environment-wide association study when he was in grad school. And this is our work now. This is work with Chirag and with Johnny Ioannidis, where this is, I'd say, in some sense, our two-dimensional generalization of this, where an EWAS considers a single phenotype at a time. If we're interested in 100 different phenotypes or 200 different phenotypes, and we'd like to scan for the relationship between that broad set of exposures with phenotypes, we're developing this approach right now. And so I'll summarize this quickly. 510 exposures, 158 clinical trait phenotypes across 8 surveys of Haynes, and then meta-analysis for those individual exposure phenotype relationships across the cohorts. This yields approximately 70,000 associations that we can look through, and then we can correct for multiplicity essentially across this entire space of associations that we look at. Long story short, a fifth of these, so nearly 20,000 in the full analysis, a little bit less in this slide right here, are significant at false discovery rate less than 5% in our adjusted models. But these effects tend to be extremely small. So they're 1.01, 1.001 type odds ratios. So I think something that limits this direct application of the genomics analogy or the GWAS analogy in looking at environmental exposures is this combination of model misspecification that we've talked about, confounding, and correlation, dense correlation amongst these individual exposures. So what you see here are QQ plots, which come out of a few different published EWAS papers. And if under the hypothesis that, under the null essentially that most of these exposures follow the diagonal here, we would expect, if we address for confounding correctly, that most of the individual hypotheses would kind of be along this line. And so this is what we see in a GWAS, for example. If we don't correct for stratification, we typically see QQ plots that look like this. And in published experiments, pretty much no matter how we adjust, we see this extensive departure of our test statistics from the null here. Okay, so quickly, the second main comparison that I'll make is to physics. So I think there are some fantastic examples of large team science, and specifically also the tailoring of evidentiary standards that has led to some real gains that I think have lessons that we can translate to exposure science. Does anyone recognize what this little blip is here in this plot? So you probably saw the news around this. It was all over the world. So this is the Higgs boson. That's the evidence for the Higgs boson from the Large Hadron Collider a few years ago. It was all over the news. This is the proof of the existence of that particle, and there was a lot of coverage. And Scientific American had a nice article about this discovery, and it was called 5 sigma. What's that? It mentioned that the evidentiary standards, the p-value threshold, was 5 sigma for this discovery. And so I thought that was pretty interesting. Typically when we're publishing studies in environmental health or in medicine and human health, 2 sigma or 0.05 is the threshold that we use. So why in particle physics is it 5 sigma, and why in most of human health and environmental epidemiology is it 2 sigma? And so I think we can discuss this. There might be good reason for this. When I chat with particle physicists about why we need to set evidentiary thresholds this rigid, the response, which I think makes quite a bit of sense, is that these discoveries are so punctuating in the field of physics that decades of work follow on their basis. But typically in medicine, human health, environmental science, we're not claiming discoveries that then derivative work follows for decades. So no individual discovery is so critical that we need to set an evidentiary threshold this high. So I don't know if that's true. I think you could make a good argument that a randomized controlled trial that's going to determine policy for statins might require a more rigid evidentiary threshold than we typically use. So the other, and I'm almost out of time, so the other main comparison that I thought was pretty interesting, and this is largely because we don't really take advantage of these methods, I think, in our field, is this idea in physics of blinding the analyst from the actual data itself. And so there's many ways to do this, but adding a little bit of noise to the actual results that then is held out, and the analysis, the calibration of the errors, the understanding of variance, Monte Carlo simulations, things that we need to do to make sure that the experiment is working can all be done in the absence of the actual values themselves, and only right before sort of publication or release can we actually blind or unblind the analysis. So quickly in summary, I think many of the central challenges of applying AI, environmental health, and exposure science are not uniquely AI challenges. They're data challenges. Environmental data are very complex. Many of us appreciate. I think there's a couple of potential solutions, new measurement platforms, large consortia, team science that have been used very successfully in genomics and physics. Analytic choices, multiplicity, so potential solutions, we didn't talk about it, but we can discuss pre-registration, blinding, agnostic, wide association study approaches. And then another issue that I think is very relevant, and I'd love to see discussion of today, is that there's extreme missingness in a lot of this data. This really hampers our own work. I would love for an imputation method just to solve all these problems, and for us not to have to invent ways of doing this, or come up with different types of reproducibility criterions. But I think that's also going to be critical. And then finally, useful comparisons in high throughput genomics and communal science and physics. All right, thank you. We have time later. The questions?
What I'm going to ask is if there's any burning questions now. Any burning questions? Yep, we've got one.
Okay.
Yeah, I didn't know whether you can call it technically burning, but I wanted to ask you about the QQ plots that you showed, the 4 QQ plots.
Yeah.
So was that taken from the study that you did for phenome by environment, like 510?
No, they're from 3 separate EWAS. So one is from the investigation of all drugs for association with a set of quantitative outcomes, and then the other is from a telomere EWAS that we published a few years ago. So those are from 3 entirely separate applications that have been published, that are essentially published EWAS and a drug loss.
So what is the next step? That's my question, because we already probably knew this before running the analysis, that this is how this is going to look like.
So I think our perspective in the paper, and I think now still this is our perspective, is that we don't have good ways of actually quantifying how misspecified our models are typically in these settings. So in genomics, what we would do is we would use a genomic inflation factor, and we would take the median of either the p-values or the chi-squared statistics and divide them by the median of the test statistics under the null. So what we proposed in this paper is a similar x-wide inflation factor, which is the median of the test statistics from an EWAS divided by the median of the test statistics under the null. And so in genomics, the rule of thumb is 1.05, 1.1 are acceptable levels. Right now, we're in the 2.5, 3, 3.5 range for this similar x-wide inflation factor in environmental exposure studies. So our perspective is, first, let's formalize, because I think there's still work. This is the tip of the iceberg and what we can do in trying to actually, I'd say, attack this problem. But in general, my view is that we should first formalize the measurement of it, and then we can think about how to address confounding a little bit better. So, I hope that answers the question, or.
I think I'll let you move on.
Okay. All right.
The session
Thank you.
yeah. Thank you.